your slide, and that's mine. So what I'm going to talk about for the first half hour, and then I'll pass the torch on to Renata, and who's next? And so what I want to talk about is our work, which is kind of loose title of chemical biology of adventitious rooting. And what I want to do is just to walk you through a kind of approach that we're doing to solve an age-old mystery, which is very important to hazelnut biology. And if you look at auxin research, it sort of has a history that starts early. I think when Dawn was a child, 1750 or so. And
Year before.
and has progressed as science and technologies have evolved. It's progressed along with it, and chemical biology is an approach that's kind of a rehash of inhibitor studies that goes back to the biochemistry days, the 1930s, but it's taken on some new directions by the ability to screen massive amounts of things against very specific biochemical targets. And so it's, like everything in science, it's a cycle. But I just wanted to show you this because it shows the different kinds of approaches that are possible and the kinds of ways you can deal with things. And it puts what we're doing in context of that cycle. And so chemical biology gives you a lot of tools, and those tools allow you to approach a fairly large number of critical issues, large chemical screening by molecules that have really unique properties. Sometimes with small molecules, you can target reactions which are lethal in the form of mutants, so that you can't approach them with mutants, or situations like we work on where we have the lethality issue, but we also have multi-copy genes. And sometimes those go hand in hand because a lot of times in biology, if something is so critical that it's going to kill the plant if you don't have it, the genetics of the plant favors having multiple copies of those critical genes. And so as you begin to approach developmental events that are critical, especially through embryogenesis or other critical life stages, you will often find that you approach those same problems. You can't do them with the classic, as molecular biologists call it, wreck and check, spray and away, Dawn stay, wreck and check. So you can't use that, but you have to try some other approach which gets you further along in those. And so that's kind of the position we're in. The model system that we use when we're not working on hazelnut, although most of our work these days involves both, is one where we're looking at adventitious rooting. Arabidopsis is nice because a light signal induces adventitious rooting on the hypocotyl. So we've gotten a lot of mileage out of using that very simple system. That light switch actually turns out to work quite fine in hazelnut. Recent paper last year on using light and dark periods. Which works as well. So there is a light signal that's involved in the adventitious rooting. But if you actually look at adventitious rooting historically, you will find that it dates back into the mid-1930s, 1934 to be exact, where a large number of different auxin-like compounds were screened for induction of roots. If you actually look at induction of adventitious rooting, it goes back to pre-biblical times, but as a matter of fact, it's associated with the conversion of mankind from a nomadic peoples to a fixed base agriculture, because you can grow wheat any place, anywhere you want. You just have to be there at the start of the season and grow it, and then you can pick up and go anywhere. But if you grow fruit trees, you better stay around a while. And so it's played an important role in our human history. But endobutyric acid was found in the 1930s. The group at Cornell that originally published that was important, and companies evolved that were selling IPA. You can go to Walmart right now and buy it for 5 times what it's worth. And it's the standard treatment that some people use NAA as well, but mostly it's IBA and its various forms. And it's persisted. This book from the 1940s by Kenneth Hamann is a classic where they basically catalog all the different things that you can use it for. Now UC Davis has a website and you can go, it's been active as a website for the last 25 years. And you can basically type in the species you're interested in and it will tell you all the different methods that you can use IBA for. And that's, so that's an old technology. It's a very old technology, but it's the same one that people now use all the time and they don't use anything else. So what I want to talk about is how you might get past this old technology. I have students that whose parents weren't born when IBA started to be used. And sometimes their grandparents hadn't been born when IBA was started to be used. It's a long, old technique. And this is what it looks like if you want to go to the store. You can pick any one of those. There's all kinds of papers about which one works better. They all look the same. IBA occurs naturally in plants. When it was originally reported, it was always on the list of synthetic auxins. For my PhD work, I developed a method for quantitating IAA using IBA as a standard. And my friend Frank Epstein was using that method in Israel, and he got a really weird result, which turned out to be that there was IBA in the plant. And together we identified that and published good mass spectral data identifying IBA as a natural compound. I became the darling of the entire IBA industry. All those companies that I showed you on the previous slide, several of them are Swiss companies. I got boxes of Swiss chocolates in the mail because I saved them all the cost of registering it through the EPA at the time, and they were very happy with me. But I couldn't really take the chocolate because I worked for USDA at the time. We couldn't take gifts, so I had to put it out and have everybody in my lab eat it, and that was good for morale.
So are you saying that they didn't, because it proved that it was naturally occurring, they didn't have to register it as a...
It is synthetic.
Okay.
Yeah. IBA technically is not an auxin, and I'll show you a little bit of data on this later. It's an auxin precursor. It's also looks like it's made from indole acetic acid, but it also goes to indole acetic acid where it's active. And IBA conversion is necessary, but not always sufficient to induce rooting when lines are difficult to root. IBA, like other auxins, it moves around the plant, but unlike IAA, which when it moves around the plant pretty much stays as IAA, IBA is conjugated and heavily conjugated as it moves precipitally in a plant. And we showed this a number of years ago, and it just shows here that and conjugation plays a major part in its metabolism.
What's the primary conjugate? Oh, there it is.
Oh, there you go. Right. But IBA, in order to be active, it has to go through beta oxidation. And I know that all of you remember beta oxidation from your high school biology class. Beta oxidation is critically important in all organisms, in that you have to be able to take fatty acids and move them into acetyl-CoA. And in order to do that, the peroxisome, an organelle that 50% of plant biologists have never heard of, the peroxisome does that process. And many of the enzymes involved in fatty acid beta oxidation are also involved in IBA, beta oxidation, but there are specific ones that are unique to IBA. Okay. And if you look, through a genetic screen, for those of you who are plant breeders and you don't believe anything unless you can see a diagram like this, the important part of this, though, is the biochemistry. That what happens is if you knock out these genes, the plants become resistant to high levels of IBA. because they simply can't do the beta oxidation. And if you look at that in more detail, what you find is that there's a number of those genes that if you knock them out, plants become resistant to IPA. Or as we say, since we're trying to get them to root by IPA, they're not responsive. However you like to see it, but you can see that there are quite a few genes that are involved in that process. I did this work with colleague Lucia Strader, and I think that pretty well defines the fact that IBA is not itself an auxin, although there are still some people who claim, without doing the kind of good mutant analysis we did, that IBA has some unique characters, but they're not related to adventitious rooting. So in terms of adventitious rooting, that conversion is critically important. If you look and you compare rooting to the ability to convert IBA to IAA, this Molly Tillman did when she was a graduate student with Dawn and I. This is actually not in Hazelnut, this is in Elm, but it's the same general lesson. It looks better in Elm because it just does. But what you see is that if you have good conversion to IAA, that the rooting is somewhat better, and that's just, look at the top graph and the bottom graph and compare the two. But it's not the whole story, because some that do the conversion quite well do not root that well, and some that don't. But in general, the ability to do the conversion maps to, if you can't do the conversion, you can't form the root. But if you can do the conversion, you may still not form a root. So it's not the full answer. Okay, so this becomes complicated. And complicated areas of biochemistry are the kinds of things that I do well and I love. And it drives most of the people who are listening to my talks crazy. But nonetheless, we're going to go through this. Basically, if you apply IBA, it goes through beta oxidation, forms a bioactive auxin. That bioactive auxin has several fates. The applied IBA has several fates. It can be conjugated directly as IBA conjugates. It can go to IAA and be conjugated. It can also, bioactive auxin can have IAA de novo synthesis as well during that time. And it can then also go on to conjugates, which themselves are oxidized, and they can be, bioactive auxin can be oxidized directly itself. I think I got everything, right? Okay, so it's complicated, but it's a finite number of things. So the question is, with this complicated array, what can we do to try to improve on just feeding IBA? Because if you look, you're feeding IBA, that's one arrow, right? But there's lots of arrows. And when a complex system, the more things that you can attack or can affect, the greater chance you have of success. Okay. And so there are tools. And it said chemical biology, there are tools. So the first inhibitor of conjugation, amino acid conjugation that came out was AIEP. I'm not going to give you the whole name. And that I'm going to talk a little bit about how it works. And then I'm going to give you a really quick introduction to two compounds that have come out within the last 30 months or so. 22 months or so. KKI and Nallicin, which are also inhibitors of that process. We know more about the biology of AIEP, but there's a terrible story, which I'm not going to tell today, about how hard it is to get that stuff. But then we'll talk about the others. And then Nexilin and YDF and Epicin and PEM, but carbimazines are all other inhibitors that we're not going to push too much to go. But, okay, so this comes from a paper looking at carnation, and I'm not a real fan of carnations, but I'm a fan of this paper. This was a paper where they actually looked at difficult and easy to root, and the thing you want to remember is 203R8 is difficult to root, and the other two are relatively easier. You remember that? Yeah, okay, you need to know that. Okay, and what you find is that auxin conjugation is greater in the one that is difficult to root, and that's the red line. And the IA levels are lower. And this just shows a scale of auxin-induced gene expression in the different lines. And it shows the GH3 conjugating activity. And it shows, what did you say? And this is just a basic idea of what happens if you give the inhibitor, and with the difficult to root, you actually get a lot more roots. So if you inhibit conjugation, it doesn't have much of an effect on those that root well. Luckily in hazelnut, we don't have anything that roots well. We have difficult to root and very difficult to root.
Yeah.
Yeah. So we don't know. We're just starting these experiments now. Okay, the story with AIEP is that it's very difficult to make it, and it's biologically not very stable because it's an analog of the intermediate in the pathway. And I worked for 2 years trying to get it and make it myself. It's not really easy. But within the last little bit, both KKI and allicin have come around. And the nice thing about them is if you look at the inhibitor constant on these, you'll see that in AIEP, it's measured in micromolars, 10th of a micromolar to micromolar. And the allicin and the KKI are considerably higher KIs, so they're much more inhibitory. There's only 2 places in the world that you can get these. Allicin, the original author, is in Japan, and I had it made. I only have 13 grams of it, which, given that it's its inhibitor constant is in the nanomolar range, is quite a lot. We can inhibit a lot of conjugation. KKI, but likewise, we have something on the order of 15 grams. So we've stocked up on these. Now you can ask, why do you buy so much? And it's terms that it's actually much cheaper to buy a lot than it is to buy a little. I mean, it costs you more, but per experiment. So we have enough to do quite a lot of experiments with these, and that's kind of an investment in faith because we haven't tried these yet. I guess
See progress.
progress, yes.
How much does it cost you?
Huh?
How much does it cost?
KKI, I don't remember. Allicin, it costs initially $5,500, and then they made for 10 grams, but then they end up making 13 and a half. I think I had to kick in another $1,200 or something.
Plus, I think.
Nothing. Well, yeah, grant money, it's, yeah, it's-
Well, I was thinking maybe $1 million, right?
no.
That's what I mean, yeah.
No, but we have lots of compounds, so we.
Yeah, yeah.
But anyway,
Where do you store it? It's safe?
I'm not gonna tell you. I'm gonna tell you those first. So anyway, it's just, so the KIs are the important part, and so we just, okay, we're good. I haven't lost anyone yet. All right, so we're back to this diagram, and it's just an example of the fact. So there's maxillin, which actually we haven't done a lot of work with yet, but it actually increases the rate of beta oxidation of IBA. And so we have that on the books for trying. It's an interesting compound. It was discovered in my lab, but I'm not an author on the paper because it was done by my students. And that's the way professors should run their labs. We did it a long time ago before we actually became interested in this part of the project. So I kind of rediscovered it actually. And then the 2 compounds that we were just talking about, they inhibit this, but they also likely inhibit this as well. And so it's pretty interesting. Then we also have the inhibitors that were on that original slide, which we acquired the same weight on, at about the same prices, which inhibit IA biosynthesis. And this, you can read about that in phytochemistry. Molly Cohen's paper that came out last year is a really good example of how to use those effectively. In addition to in addition to those compounds, an interesting one, and I talked about this before in this forum, that there's also things like 4-chloro IBA. And the odd thing about this is that apparently they go through beta oxidation just fine through the same pathway. It looks like the same pathway that IBA does. And so putting halogenation groups on there. What are you looking at?
I'm looking at the structure.
Anyway, so we have, and then, so all of these can be done with the inhibitor of conjugation as well as the using IBA itself.
What was the rationale for using an inhibitor of IAA by biosynthesis?
Oh, there are some cases where it's thought that the amount of the IA in the tissue might actually inhibit adventitious rooting. And therefore, if we can drop the levels of IA, the endogenous IA, then what we feed it will become dominant.
Gotcha. Thank you.
We haven't done that experiment. But that's the rationale.
Is there an expected starting range of IBA that would get to that result?
Well, we don't,
General question.
yeah, so one of the important questions that we are answering, and I'll get to this in a minute, would be to actually know what the endogenous players are in terms of IA levels that we start in the different plants, and what happens when you put IBA on it, and using the treatment we normally use, but then modifying that treatment. And we can do that. We just haven't had the personnel basically. But let's see. So I always like to nudge Dawn a little bit about the Forever Green funding process, since I'm not funded by Forever Green. And so the important thing here is that this whole project I just talked about was presented in 2021 for Forever Green funding. And in the infinite wisdom of the Forever Green funding thing, they decided that it wasn't really worthy of Forever Green funding. And that's kind of sad, except I can't tell you the results of this because USDA won't let me tell you. But I think you can gather from this what the result might be. So instead of getting my $150,000 from Forever Green, I got 650. I might get $650,000 from USDA to do exactly the same thing that wasn't apparently worthy of funding from our program. I think it's,
So you lucked out again.
yeah. I just think it's really important that we not leave those things, you know, to the whims of fate and the whims of college administrators if they're not doing a good job. I just, you know, it's not sour grapes because I got money. But, well, maybe I have. I can't tell you. But I think it is a good lesson for us to consider. You know, I used to be a grants administrator, so I look at these things with due diligence, right? Right?
Yeah, I won't say anything.
Right, so. Anyway, that's all I really have to say. I still put Forever Green up there because they funded the first part of our work. I didn't want to take it down, and Mitch would be mad at me. So, so we can answer questions. Well, we did.
Les Everett asked on the chat, what is the availability of NSF funding for this basic work?
Oh, we are funded for we're funded for a spin-off kind of thing. So we look at tryptophan metabolism in general. And so we just got funding from NSF, for a large grant. That one I can talk about. So we got $1.3 million to study the conversion of tryptophan to niacin, which is an age-old problem in plant biology. So we're working on that. We have started getting ready to submit is one on oxen metabolism, which would actually be consistent with that. We've been funded by them.
What's your gut feeling? You know, obviously this rooting work has been going on for. You know, that one you showed there was in 1947. That was the year I was born. So this stuff has been going on and. Give us a little summary as to what you think. Is the underlying reason that some things root. Even within the same species. And other lines don't. What is your underlying rationale for why in the hell that is?
Yeah, so that's a, it's actually, okay, let's back up and let's just say that first of all, it's amazing that adventitious rooting occurs, right? I mean, it's kind of like the lizard that regenerates its tail. You know, unfortunately, we don't regenerate our tails, but lizards do, right? And the question is, you know, how do you have this induction of a new organ? So organogenesis is complicated. But we know that oxen is involved in it because it's part of a wound sensing apparatus, the plant. And why is it part of the wound sensing apparatus? It's because the most important part of dealing with a wound in a plant is regenerating vasculature.
Yeah.
Right. And so part of that process of wound regeneration is producing new roots. Because injury is going to induce the need for new roots as well as the need for regenerating the vascular connections that are required. Why don't some do it? It's harder to envision why, but a lot of trees don't have as nearly as imperative of a reason for doing that as some other plants might. But certainly they've been doing it for a long time because, you know, you can read, well, I think I have a review I'm writing right now where I have quotations from the Old Testament and things that go back into the Greek scholars that talk about the problems of raising fruit trees from seeds, or what they call degeneration, which is the fact that seeds continually propagated from fruit trees end up with fruit trees that aren't that good. So cuttings and propagate has a history that goes back to early human civilization. So it's certainly, and most of the trees that have a long history in human civilization root without any IBA, because IBA was only in 1934. But sometimes they stimulated it by adding seeds of grain and things like that into the trench that they dug to plant the cutting, because endosperm has a lot of conjugates. So I don't know exactly why, but you know, as a basic biologist, I think the more we know about the biochemistry of what we're applying, the greater chances of figuring it out.
When you go back and you look at the ones that root versus the ones that don't, and do a whole genome analysis, what do you see?
You know, we haven't done that.
I understand.
Are you talking about hazels or like different species?
No, I'm just talking in general, right, in terms of. It could be applied to hazels or any damn thing. Has anyone done the metabolomics approach? Looking at gene response to these molecules that are probably turning on damn genes.
I think I
right?
showed you the best example of that in terms of the Carnation project.
Yeah,
But there aren't that many good systems for easy and difficult to root plant systems. And in my career,
I mean, yes, no types.
in my career, I've done olive when I was working with the Israelis.
Yeah.
Because they had that system. When I was in Italy, I used pear because they had ones that in the micropropagation system worked better and worse. And now I have hazelnut. So I've had a long career. I found 3. But they all have the same problem. is that difficult to root or hard to get in large numbers. I wonder why. So it's, so that, I think that actually is a good approach, and probably carnation is actually one where you could actually,
Yeah, but in the hazelnuts, you have, a pretty good differential, don't you, in some of the lines?
yes,
It's, you have lines that are no. Or damn close to no,
damn close to no.
yeah. And you have others that are. Okay,
So we have some that are.
can't we
pretty
take,
good.
couldn't we take? This metabolite genomic approach,
We could take a metabolomics approach. Genomics would be much more difficult.
I understand,
I think that's kind of the thing that I'm actually funded to do that now. No, I might be funded to do.
So that kind of idea is included in that new funding.
Well, at the biochemical, at the metabolomics level, but not at the.
I got you.
Not
Okay.
at the.
So last week, Devin Peterson said that, some of the flavor compounds, and I think, if my memory is correct, I think they were bitter flavor compounds that they've identified in hazelnut kernels are actually auxins.
No, they're actually,
They're built off the
They're-
auxin pathway.
well, no, let's see. So you see, you see the oxi compounds up there. So his are all oxi, they're different glycosides of oxi.
Okay, so does that mean they're precursors?
That means that they're dead end products.
End products. Tertiary metabolites.
Oh, okay,
Right, so that's that, so, but they're really interesting compounds.
okay.
To us, they're interesting compounds because they're new metabolites that log into that sugar conjugate on a top.
Mhm.
Could they be feedback inhibitors?
That would be a stretch to know. No one's looked at that.
Should we be worried that our varieties that root more easily are more likely to have those compounds in the nuts? Or are there like
Is that true?
47 steps?
Is that true?
What?
The ones that are bitter, root bitter?
When we looked at. Devin followed up and sent the graph with which ones they used. There's only out of the 12 of our varieties, there's only one that's really of high interest. And it's one that roots well and had really good flavor.
Which one?
Or 7-1.
The same that I.
Yeah, but those other varieties, we don't know very much about because they haven't been.
Yeah, I don't think that you can extrapolate easily from a stem to a nut.
Great.
But don't hold me to that 10 years from now.
Well, we know now we know it's, something that
But
can be tested for with, new.
You see,
promising
I
varieties,
finished on
but-
time, but now you guys are making.
well, the whole purpose of
That's
this.
good.
So,
Yeah, but
but
he's
Ted
next
is
to me. He's gonna
doing
keep
some
that.
research.
Huh?
As long as we've got 90 minutes, I think we're good.
Tang is working on some research that involves kernel, correct?
Yes, we're looking, okay, so we want to identify. It's part of this process. That's a good question, but none of the people online know. So we want to find out what the auxin conjugates are in hazelnut. We look in the stem, it's a little bit difficult to find them because they're at much lower levels. So we thought we would look at the nuts themselves, and so we are looking at that. Because that's the road to identifying them. And why do we want to do that? We want to do that because if we know what the conjugates are, we can do a couple of things. First of all, I just showed you, we can know how to stop them from forming. But if we know what they are, because of the way that's inhibited, we can stop them from being hydrolyzed as well. And there might be some advantage to being able to do both of those things. So we want to know what they are, because the best way to fight a problem is to know who the enemy is. I'm partly Ukrainian, so. So but we have to, we have to know what the components are in that system, because what we're really trying to do is map out all of these processes so that we can manipulate them and functional way. So that's why we're looking at the kernels. It's just because I thought that was faster. Once we identify something, finding it even at low levels is actually pretty easy. But not knowing what you're looking for is searching for new, like, basically de novo identification is much more difficult than something that's at low levels than at higher levels.
How much easier is it to root Avalana? Is it 100%? Is that a yes, no?
It's not 100%.
It's not,
Easier on average.
it's
I understand,
easier
but
than
is it-
ours, but I visited North American Plants, which is a major plant propagation facility in Oregon. And they had buildings full of all sorts of species that looked great. Then we walked into the room where they were rooting Avalana. It was like, oh, these don't look very good at all.
If you read papers on the hazel, cuttings and propagation research and microculture stuff, they'll say like, yeah, there's a bunch that doesn't come through tissue culture.
But the rooms that were, or the greenhouses that were, what varieties or cultivars were those?
They were commercial cultivars from Oregon State University.
Well, other species.
With a lot of Avalana in them, though.
They were pure Avalana.
I think the
No,
Yeah,
I mean,
missed
they were,
I missed what you just said there.
so
Propagating Avalana.
compared to,
the hybrids.
I don't remember what other
Birches and
crop.
blueberries.
Birches and
yeah, stuff.
blueberries and apricots and pecans and so forth.
Well, make sure I understand what you just said though. So you walked into a room of Avalana, period. They didn't look good,
And they didn't look good compared to the rest of the greenhouse,
which was other species.
which is other
Yeah,
species.
yeah,
But one of the,
yeah.
one of the problems you have when you work in micropropagation is all of the papers that get published are on the things that work really well. And everybody expects that, that it can be done, because in some, if you get the right, if you
Don't
get the
publish
right,
disasters.
if you get the right line and the wind is blowing in the right direction and it's a good afternoon, you can get really good results in various systems.
That's a very good point to bring up. And
Because,
in terms of the scientific world,
yeah,
that's a huge flaw.
yes,
That
and
people don't publish
the stuff
what didn't work.
what didn't work.
Yeah.
And they don't publish that their method didn't work with the other 85 lines that they have.
That's
They just publish what they got.
right. It's a weakness. It's a huge weakness in
But-
science.
But the challenge that Dawn gave me was to have a method that worked for everything, because they want to pick the plants that work the best in Minnesota farms. And they don't want to be limited in the taste, quality, and the disease resistance by propagation. And so what the goal for us is to get propagation off the table as a roadblock or generating these other things, so you can focus on useful things like, does it taste good and is the nut really huge? Right, because that should be what's on the table. So we have a lot of different things, challenges. And I think that, if to answer Don's question that he posed, it's likely that we will be able to use the oxen studies to get past many of those barriers. Will we get past them all? I doubt it, because, you saw the elm data that some things convert quite well, and yet, now we don't know, maybe they conjugate it well after, we don't know all those details. But I think it's simple-minded to assume that organogenesis is only an oxen-regulated phenomenon and there's nothing else involved.
Should we pass it on to the next speaker at this point?
You're the boss.
You all know how much time you need, so.
Yeah.
Seems like maybe that's a good moment.
Yes,
Okay, great.
and it's giving me a notice.
I'm up next.
No,
Oh, I
I'm
didn't
saying
say.
if we.
Oh, I see. Okay. Gotcha.
You can be if you want.
No. I didn't say it.
You just do, you should come up.
They're beautiful.
Have you been introduced?
Introduce yourself.
So yes, I'm Renata, I work in Jerry's going on. So, and I work with hazelnuts. Okay, so I work with micropropagation. And I think my talk is gonna be super quick because Jerry covered everything. And in this conversation, I think you guys always also.
So just show us the ones with the big roots. That's all we want. That's all we care.
Just show some
This stuff
pictures.
is gibberish. Now show us the roots.
Okay. So, as everybody knows, so we are working with hybrid hazelnuts that could be a potential perennial crop for Minnesota. So the hybrids combine the nut quality and yield from the European and cold hardness and disease resistance from the American hazelnuts. So this is an overview about how micropropagation works. So we collect buds from greenhouse, or if you don't have a greenhouse, we can collect from field. But we collect axillary buds from greenhouse and then put in a media to start grow some leaves. Once we have leaves, we transfer for shoot elongation. And then after that, we can do some multiplication or replication and use this, and use this stage to start our experiments. For example, to try induced roots. That should be the next step. We can use these plantlets with at least 4 leaves. So, and then we can induce roots. And then later transfer for acclimatization in the greenhouse, and probably later for field. But this is the beautiful story. But for hybrid hazelnuts, we are locked in number 6. So to do all this process starting on the number 2, we need to, let's see, next slide. Okay, I'm gonna count down and follow the slides. So micropropagation is an alternative method to seed or vegetative propagation, but we still facing some barriers to commercialize hybrid hazelnuts. So the 3 main roadblocks that we have is how to establish the crop, and how to improve the process of rooting and later acclimatization. So I think we are doing well in the first 2 roadblocks. So last year on September, we published this communication that we found at least 2 sterilization, efficient sterilization protocol for hazelnuts. So we tested these 2 protocols in almost all our genotypes, even collect from field or greenhouse. We had a really success to sterilize these materials to put in tissue culture later. So, and then we also, after that, we try to induce roots in these different genotypes. So here we choose 2 that is considered easy and difficult to root, and we apply IBA. And also after IBA, we follow treatment with dark period of 7 days of dark. And after this darkness treatment, we move the plants for light for 24 hours. Light, they was in a chamber with 24 hours light. And then we start to see some roots growing after between 12 and 14 days, we can see the roots, visible roots growing. So you can see here, this is the genotype Hamann that is considered easy to root. We saw visible roots at 12 days after transition to 24 hours light. And then now we adopted this as a protocol. So for our experiment, so every time that we apply some compound to induce root, we keep the plants on the dark for 7 days. And then after that, we move them for a chamber with 24 hours light. So figures B is shoot with 30 days after transfer. So you can see in the bottom, there's a lot of roots. And I would say that in this stage of roots, we can probably move for substrate. And if you want to do acclimatization in the greenhouse, it's ready to go. But it's not our focus. Our focus is get these plants to do other experiments so we can replicate these plants and do other trials. So as Jerry told, the root induction responses are not quite uniform between the genotypes. So either for micropropagation, and Lois can tell also for vegetative propagation, right? It's difficult. So, and then we can classify the hybrids in 2 ways: the easiest to root and difficult to root. So, as Jerry also mentioned, we tested a lot of compounds. We are using IPA as our standard. And then here is all the compounds that was already tested. And Nelson, KKI, and the sorting is in progress. So some of these compounds was tested in different plants. We have tomatoes. We have also this N-phenyl IBA and phenyl IBA. We have some students in our lab that prepared these compounds, and they are developed to use by USDA in the forest service to root some trees. So for all of these compounds tested, we had some success, but compared with IBA, we still need to adjust, to optimize some rates, some concentrations. Unfortunately, sometimes we don't have enough material. Sometimes we have the compound, but the plants enter in dormancy. We need to wait a little bit to get more materials. And also the lack of people to help us in the lab. So, but if you want to see pictures, we have some. If you want to see roots. So here are some examples from how the structure of roots looks like. So this is when we apply IBA, it's beautiful with vigorous 5,6-dichloride IAA. So here we have 4-chloral.
You do want to tell them that not every seedling looks like that.
Oh yeah, we have for each experiment, usually we do like 10 repetitions, and then based on the repetitions, we just see like the percentage of roots for each treatment. So I just took the picture that looks good. Yeah, but you can see here, for example, the phenyl IBA. Another thing that I can bring out that maybe influence in rooting, it's the season that you apply the. If it's fall, for example, even if they are in the controlled environment, they kind of know that it's fall, for example. You can see in this one, that there is kind of one leaf. This picture is not good, but I don't know, all these other experiments I did actually this was in February, was kind of winter time, but I feel that the plants know the seasons, like.
So they do better in the spring.
Yes, every experiment that I did, I got better results in like spring.
So I'm wondering about the leaves.
Mhm.
Those leaves don't look particularly healthy. Are they
Yeah.
going to, is the plant going to survive in spite of that?
Yeah, so these brown ones, are you talking about?
Yeah,
Yeah, sometimes they touch the media and they oxidize.
okay.
Yeah, because it's humidity environment. And they are in contact of the media here and then they just turn brown.
They'll get new leaves.
Yeah, they will,
okay.
they
Yeah.
will. Mhm.
You just have to wait.
So bud
Because-
activity is not inhibited, because.
No.
Much about IBA inhibiting bud.
So usually after this phase, for example, they get roots. I wait like probably 1 or 2 months and then I transfer for another media with zeatin that I can grow shoots or new leaves again. Okay, so next experiments will be with these 3 other compounds, and pretty soon we are gonna have some results. And we are the next step also, we are gonna play more with another range of. And what else? And we are doing with Tian, we are doing conjugates, right? Metabolomic analysis. Probably in the next meeting, we are gonna have some results also about the conjugates.
Analysis goes a lot faster with tissue culture.
Yeah,
So why can't you prepare your plant material so it's all uniform and run the experiments with uniform plant material? You see what I mean in terms of, it sounds like it's hit and miss in terms of growing your plant materials, and then test each of these compounds against uniform plant material.
oh yeah, so every time, every experiment that I do, the
Yeah,
plants have the same age.
I see.
Yeah, I have like a batch of a specific month, and
It's,
then,
it's my fault, Don, because when I buy these compounds, I take them whenever the chemists
I
send them.
understand, yeah.
yeah, but
And she
the,
complains that I don't wait until the springtime to get her.
yeah.
I see.
But at least the plant material that I work belongs to the same batch, the same age.
I see.
I take care of this because I can't work with kind of, some plants look like with hard tissue,
Yeah.
other ones soft tissue. I can't mix it up. Yeah,
Because what you're starting with when you're trying to root are multiplications of what you originally multiplied in vitro, right? So those should be very uniform.
yeah. Yeah,
I guess I had a similar question of, is there a way to, I assume the spring, fall, or spring other seasons issues that the plants are entering some kind of dormancy. So can you break that with growth chamber or some kind of period of treatment so
Yeah,
that
we-
they all go through it and
yeah, we
Yeah.
have a treat, we
We-
call, go ahead.
Yeah. We have to have that, so we found there's a minimum 6-week cold treatment, all of our plants go through staggered 6-week cold treatments.
Okay.
Sometime starting in late fall through the winter. And we also can break it in additionally by adding a GA4. But that was a preview, that was before you came.
And so it's still, despite that, it sounds like there's still some
Yeah, we
seasonality to it.
still have to do this chilling treatment.
So out of all of the compounds that we've been hearing about, there is no silver bullet
Yeah,
that's arrived yet, right?
well,
Or is it due to the fact that you haven't had enough time to work with concentrations, combinations, and
That's
all
true
of those
too.
things?
Yeah.
Yeah,
yeah, if you looked at her, but some of the strong oxen type compounds give almost like an ethylene effect, suggesting maybe that the levels are too high.
that's what I was picking up on.
Yeah, and
Yeah.
so we had this discussion yesterday. She said, well, that they failed. And I said, no, they didn't fail.
Well,
They
that's
just didn't
what I-
give you anything.
that's
better
what I
than
wanted.
that.
Yeah, that's what I was asking whether we knew enough about. Well, we went through that whole array, period of time, looking at various combinations to regenerate corn from tissue culture. It took years, and it was a matter of coming up with the right mixtures, right combinations that worked on a few lines, but didn't work on all. And I think there's been some advancements over the years that works on more and more of the genetic material, but the original lines only worked on 2.
Well, anyway, the strong oxens, I think, we can't say that they work
Yeah,
better than IBA, but we can say they do. At least using it at the same concentration as IBA still give you roots.
yeah.
And I don't think there's a statistical difference between the 2 compounds, but IBA is optimized because it's been used for 3 generations.
So from listening to you, it's a matter of getting more people in that lab
Mhm.
to work with you, right?
Yeah.
And to be able to handle more of the experiments.
We have 2 more speakers and we're running out of time.
And I have, and I'm fine with jumping in and doing that, if that's where you're going. I do have a couple of questions for you though. You showed the humble 16 genotype up there. Is that close or is that a more distant relative to the newest hybrids?
Let me explain what humble,
Okay,
that was a seedling. So we don't know
Oh.
what seedling it is, but we do know that it was one of our hybrids crossed with a European hazelnut.
oh, okay.
So it's got a higher percentage of. Yeah,
Well, yeah.
so we have no, until we get multiplication, grow it out in the field and get multiplied versions of it, we have no idea what the potential is.
And then on some of the leaves that you said were oxidized from touching the growth medium in there. Did you ever find in the pictures that we didn't see of all the other ones, any endophyte problems like the neck hollow limitation is basically, and my understanding is it's a bacterial endophyte, but it wasn't.
No.
We don't see it.
Don't see it
No.
at all. Okay.
No.
At least our sterilization protocol is really powerful to
Okay.
clean the plane.
And
We can't get from them what their sterilization protocol is, but we did isolate their contaminant.
Okay.
So,
and it was the protocol that was at the 2022 paper that you were.
Yeah, you can,
That is the protocol that was used for these.
yes,
That's awesome.
yeah, you
That-
can check it out.
That is like,
It's
I've read it and it's like
simple.
weeks worth of kitchen prep
Yeah, it's really
And-
simple.
and deep,
Uh-huh.
overkill from just reading it the first time. But if that's what it takes to get rid of the endophytes, then fantastic.
Yeah.
Yeah.
At least, in the
That's
beginning, when I started try some protocols, I got like 50% contamination. And then once I start doing this ones with field material, I got like 10%. And then with greenhouse material was like 100% without any contamination. And then I can work easily right now. I don't get
Wow.
any contamination.
Yeah, I wouldn't call Night Hollow any of those comments.
Well,
We presented this to the patients.
Did,
we always,
No,
I have you know, there's a, I don't want to take up all.
no,
your
no,
time.
you're fine.
I learned tissue culture originally from Tosh Marashigi. And he explained to me when I was an undergraduate, just starting out in life, that plant tissue culture people, there are two types. There are types like Tosh Marashigi and me, who actually like to explore exactly why things are happening and how they happen. And there are others who just like to check all the different formulas and see which one works for them and keep going. I will never solve that problem in my lifetime. Because Tosh Marashigi never solved it. He's smarter than me. And but you know, it's if you have a different, if you already know the answer, you can't be told what the best route would be.
Mm.
So I don't know, you know, we help them as much as we can. And they can read the papers too. So if you read it, I'm very happy you read it because, you know, the average paper is only read by 3 people and Renata and I read it. So at least I have it down.
See what the thought on Brent mentioned in the email, like they've taken care of the endophyte problem. Now we think we might not have enough endophytes and that's why they're not growing.
Well,
What is that?
well, Melanobacter, it does have a history of having some beneficial effects. I don't know, you know, he's doing things at a lot different lifetime scales than we are.
Yeah.
We're just kind of stuck at one thing because we're trying to solve a very specific problem. So I really don't want to compare what we're doing, but we've had things in culture a long time, and based on what he said, after a certain length of time, you start seeing those things. We don't see that. And so I don't know if that's.
That was the
because-
endophyte you isolated.
Yeah,
for him.
no, she has.
Oh, you.
I helped identify it. But because I mean, it's pink, it's kind of obvious. It's a certain class of bacteria.
So is the impact of certain endophytes on rooting known?
No.
The problem for them is that rooting is shoot growth.
Not sure.
It's shoot growth.
Yeah, I
They've
don't,
got them rooted and they're sitting there stabilized, but not.
Not growing, I see. And so. Is that understood very well in terms of endophytes' impact on shoot development? What's that literature look like? Anybody know?
in my family, my wife is the endophyte
I know.
expert, not me. But she only works on fungal endophytes.
No, I understand, I was just wondering if
All that I know is that Brent mentioned in the email of something like. You know, this is it's known that they can have some positive effects at some level.
on shoot growth.
Yeah, I think so. I think there's, you know, it's reasonable in lots of woody plants to have endophytes that have a positive effect.
Ladies and gentlemen, let's move on to our next presenters.
All right. I don't have the luxury of all of the.
I have 2 graphs that I can share my screen. It's Steve's info.
Oh, okay.
It's like the dates and the come, yeah.
But and I can do what my intention here is to do a quick backgrounder and then try to get not deep into the weeds, but try to.
Tell everybody online who you are.
So I'm Steve Unverszagt. I've been involved with hazelnuts for a couple of years now, probably.
Actually, more than that, but propagation 2 years.
Propagation, and have worked with Lois on my belly and Mark learning techniques for mound layering, interested in building the industry out. Originally, I wanted to get into this as a hazelnut farmer, like lots of people trying to get into it. Scratch the surface, and there are limitations and limitations and limitations. So I'm trying to kind of come from the backside, look at an opportunity to help propagation and work with Gerton's nursery here. And I'll give you a little bit of background on them in a second. But so my role is to help the industry with propagation volume on the top 8 selections that are in high trial levels. So that's the little background. So Gertens is wholesale plant production here in the Twin Cities. They have 23 acres of growing space under glass in greenhouse. 10 acres of that is heated floors for germination and rooting. They've got an acre of LED lights in greenhouse environments, 100 acres in ground tree production, and 150 acres worth of in container production space. So they are a huge player in all of this. Round numbers, they take several million cuttings for rooting every year. So they know the system, and I feel very comfortable sharing the opportunity to use their systems that they've put in place for their commercial operation for scaling up from research, because it seems like we're really close, and that's a pretty big motivator for me. The startup plants that we have, we started with 7 selections in 2021 and added 1 more there at 421. It was some small mound layered cuttings or plants in 2022. So we've got mother plants, maybe 10 round numbers. I'll try to kind of generalize here, so don't hold me to all of these numbers specifically. 10 mother plants as stock for canopy and shoot growth cuttings, and we've got about 100 mound layer bare root in, and all of those are in 3-foot diameter tree pots that most of us have toured Gertens facility and kind of know what the status is there. They're in those environments so that I can move them around in and out, take them to different locations, and not have hundreds of pots, and work fairly closely with a batch of cutting potential plant material. They move around from growing environments to cold storage pretty easily in that form. And we get into some stem cutting protocols here, pretty standard, straightforward. There are some variables that I'll get into going through here. Not many variables, because I'm trying to follow their standard best case commercial production practices. But there were 2 cuttings. We did one in the end of July and one in the end of August, late for cutting. And they were about 4 weeks old. The first one was selected at a 3 real leaf length. And the second one, I added 2 more leaf counts on each of those cuttings. I did the second cutting myself, and some of the Gerten staff did the first cutting. And so in working with some of my grape growing projects, the more roots that you, or I'm sorry, bud nodes that you have, the more likely it is to have rooting happen. So I added a little insurance there. And jumping ahead a little bit, we did get better propagation of roots with that second cutting in general. We measured the heights after the first cutting was 2 months old and the second cutting was a month old. Scored them at 8 inches, 4 to 8 inches, 1 to 4, and less than 1.
That is the shoot that grew after.
Yes, yeah,
Right.
and in some cases, if I had successful leads on what was originally a 4-leaf cutting,
Mhm.
I designated that was a cutting that was that tall, if in fact it had new leaves on it.
Mhm.
If it was that tall but didn't have new leaves, it got logged into a different length of category. It was kind of like the height of the newest leaf.
Mhm.
And I want to go back and add that when we do these cuttings, there are a couple different ways that they handle them based on species. They have hundreds of species that they do these perennial cuttings from. And so each of the Quonset huts are managed with robotic spray mist arms and takes all the confusion or all of the guesswork out of whatever it is, standardized things pretty easily. The standard IBA concentration was about 15, not all the way, it was 1,500 parts per million. And I wanted to find out the earlier question, if that is still too much, do you think? Because some of the comments over this last couple of
That's a pretty common.
yeah,
Mhm.
and so that's, I guess, in best practices, that's what they're using. And the question came up, is that too much? Even 1,500, even though it's common, is that too much?
In all the different trials I've done, I found somewhere between 1,000 ppm and 2,000 ppm to be best. So, and it's funny, that seems to be true even with young succulent plant material and hardwood cutting.
Okay.
And layers as well. So I'd say that probably is a good number.
Okay,
Have you checked the? I haven't done this for a while. I know I did it before, but the website from UC Davis.
no,
It does have filberts in there for,
I listed that down from my notes from what you had mentioned, so I'll jump into
if you
that.
can't find it, just send me an email.
Okay, all right.
I have a,
I know Sam McGurts was saying how he was interested in trying to go lower with IBA, just because it's, you know, if you can get the rooting, that they just help the growth.
Yep.
And like, I think we all encourage trying it
Mhm.
and seeing what it is.
There is another data point in all of this, that has to do with the length of time in IBA solution.
Mhm.
Their standard cutting protocol is to put a newly cut stem into a cold water bath, basically a liquefied bottom of a bag with an IBA concentration, and let that cool for 24 hours and absorb for 24 hours. That was their first cutting protocol. My cutting protocol, we ran into a schedule difference in when I cut and when they were going to stick it. And I thought I would have enough time for that same matching 24 hours, but they didn't. And so that second sticking was a cold 18 hours, mostly fully submerged. And not in an IBA solution. But as they were sticking them, they sprayed that same 1,500 IPA.
Submerged into cold
It's
water.
cold water.
Huh.
Yeah, to stabilize hydration and, you know, it was a closed environment, very cold. They weren't gonna metabolize and, you know, go south in that time
Was
period.
it sterile water?
I think we were always still.
Okay.
Where was I? So, when they stuck the second cuttings worth, they were only spraying 1,500 parts per million into the soil medium at the point of sticking.
So they stuck first and then they,
Put a finger hole, and as they were lowering the cutting into it, sprayed it and then sealed it with just a little pressure against it. So it was a lot less concentrated over time.
Uh-huh.
And then so, so then the hydration management sort of takes over at that point. Everything else sort of equals out from that point on. And their standard commercial spray protocol is to do a 2-minute interval between mist sprays on these robotic arms for 2 weeks, except at night, which gets into your daylight, dark light, perhaps. I'm not sure how close that is to relate that to, but at night they stop the spray mist and evening temperatures, take over and everything cools down, and then the spring or the morning hours, heat and light again, put it into another metabolic cycle. So they have, like I said, these Quonset, maybe 50 different species in a given Quonset. All of them get sprayed on the same schedule. They can take this set of rows as the boom comes down. They can do this a couple of times and pre-program. So it's a pretty robust propagation environment that they've got.
Every 2 minutes for what duration?
It's a 15 second, something like that. With, the boom will go down and back. And since these are all the species are in that same cutting stage, they all get that same 15 seconds of spray. When the boom goes back, it stops, and then 2 minutes later, 15 seconds of spray. And so they are constantly bathed in this beautiful, light, cool mist, and except for the evening hours.
And so it isn't adjusted depending on sunlight and temperature?
Not for those first 2 weeks.
Okay,
And frankly, it's never, those variables don't come into play.
Okay.
It's just within that first 2 weeks, that's the environment that these sticks are faced with. And after that 2 weeks is up, the timing on it goes to 2 hours. And it's that same, it's a 2-hour interval where the boom will come down, that same 15-second spray, stop for 2 hours, and then come back. So they ease out of the growth medium and they, push.
but they go immediately from 2 minutes to 2 hours.
I think so.
That's pretty abrupt.
So one of the upshots for this is like, these are really good results
Yes.
for the people online. This is much better than the stuff that we get with software cuttings, for sure. Particularly that they start growing again.
And Lois and I, Mark and I have had frequent discussions about the difference between mist management, MIST, and mist management. I think we need the opportunity to either overwater or intervals are wrong, or heat and concentration, humidity is, some kind of whatever. These are in open-sided plastic roofed Quonsets, and sides are rolled up, so there's ventilation, and they've been doing like I said, millions of cuttings every year, and so that's the basis for those results.
So based on spraying IBA on soil, that's gotta be indicating that a lot less concentration than
Yeah.
dipping
that's the same thing.
works, because that can't be very efficient.
So the differences between your two cutting dates could actually be differences between methods.
Yeah, there are more variables than I had hoped for between the two.
Right, right. But what it means is that there's room for more experimentation.
Yeah, and I'll get to some of those options here.
I just want to comment real quick that we think that the reason why the experiment that Mark and I didn't work was because we overmissed it.
Mhm. Yeah, and I I think that their misting cycle, especially with ambient airflow,
Mhm.
to move it off. And to allow everything to soak down, not to dry out by any means, but to soak off of the leaves, make it away from some of that oxidized,
Mhm.
And the environment in those suns is different. than
Yeah.
the
Just to
this
bring
house.
this up, so
Yeah.
there's another big meeting coming up at 2, so we have to set up for it, so we
Yeah.
have to be out of here by
Right.
probably 1:40.
I
Yeah,
will
and
jump.
there are a couple questions in the chat here.
Okay, I'll try to jump ahead here, with, standard stuff. So we then measured and put, the 8 inches and the 4 to 8 inches, and even some of the 1 to 4 inches into a 24-degree Fahrenheit, cold storage out of the 7, you know, quantity counts up there? No.
No,
So we had just under 800 total cuttings. 75% of them showed substantial rooting that could support, whether they did or not. There's probably 75% of what was showing good substantial rooting actually had this 1 inch and more new growth on top of it. So one of the bottom line statistics here or calculations was that 79% new stem and leaf growth from those cuttings that rooted. And 51%, if you go back one step further, 51% new stem and leaf growth when calculated from all cuttings. So
everything that's stuck.
everything that was stuck. Yeah.
I think, isn't that the difference between these different graphs?
Shoots.
Basically, I mean, that's,
Yeah.
Yeah.
that's
Yeah.
still reduction. It's still-
And so, the good news is that we got a lot.
Yeah.
We put them in cold storage, and those that were not tall enough to warrant cold storage at that heavily rooted stage, were left in their medium cells and went into a 39-degree greenhouse for overwintering with a bunch of the other material, mound layered bare root plants that we've gotten. Mother plants that we've gotten, all of those went into a 39-degree, give or take, greenhouse through the winter. Bad news is that 3 Bunnies also knew that we had them in that same greenhouse and did a horrendous number on the chewing through 18, 24-inch shoots that are now maybe 9 to 10 inches high. And of that 9 to 10 inches, the buds that are left are maybe half. So we Gurdon's lost at least 50% of all of this year's potential. And it just pissed me off.
Of everything.
Pretty much.
Do they have a preference for one genotype?
No, we have only good ones.
This is from the stock plants, not from the ones that were cutting this past summer.
Right, that's correct. This is all from woody, bare root plants that I had taken cuttings from, mother plants that I took canopy cuttings from, mother plants that I took basil shoot or crown shoot cuttings from. The good news is that it is a natural process for this to happen in the field. Potentially, it could allow for more side adventitious, shoots for that matter.
Was the rabbit fry pretty good?
I learned years ago that karma is a real thing, and so I delivered them to a dog park not far from, actually technically outside the dog park, but I think they're hopefully going to be happy and live where they are right now. So, so anyway, in March, we're gonna wake up all these cuttings and start cuttings. I'm sorry, wake up all the plants, all the woody plants, whatever version they're from. And starting in likely May, take again 4-week old cuttings. Once you take a cutting from a shoot, 4 weeks later, you got another cutting from that same shoot. It works miracles for the numbers. In addition to what we've got, that got us 800 cuttings in late summer. We've also got, and most of these numbers I think are right, Lois. I went through all of our mother plant contribution transplanting numbers. I've got round numbers, almost 70 mother plants. Does that sound right? Give or take. From Wasika and Rosemont that are being either plowed under or repurposed for.
It's condemned land that the plants are being rescued from.
So, bottom line, we have not a lot of previous material coming, but the mother plants that we got very good cutting volume off of,
These are like 10-year-old field plants.
are wonderful to work with.
Mhm.
I have cut some by hand, but we'll take a tree spade down to both those locations, pull up a huge amount of the root base. I have to clean them all off
Sure.
and put them into sterile, or not sterile, but a growth medium, preferred growth medium.
To protect the other plants.
Yeah,
In the nursery.
in the operation.
Yeah.
So I'm going to jump to the future possible data points aspect here, and we can take the questions in a little bit. One is, and I cannot add a lot of experimental protocols to what Gertens does commercially.
Yeah.
It's the same staff that is responsible for profit, and I'm being very conservative with what I ask them to do.
You don't want to confound their system.
Exactly.
Their system already works better. So, okay.
And so it, by default, some of this I get to do firsthand, which is really fun for me. Because then they don't have to do anything. They just tell me when they're gonna be able to move something and I'll have it done and then I'll move it. So possible data points for the future, to test out the IDA concentration range for best rooting performance, whether it's a toxicity aspect or the best conversion to IAA. If you start lower, does that give you more? So that's one. We talked to Brandon about gibberellic acid concentration range for those that are rooted but not elongated, and I have several trays, maybe a few hundred of those plants in that stage that have roots but no tops. There was
No tops are not growing.
the roots have fully formed to the point where I could have put them into the rooted cooler, but they had no tops, no stems, and the criteria for Gertens is that unless you have a 1-foot stem to winter, that plant gets destroyed in their commercial protocol. And so I'm taking that way down. I've got new leaves on a 3-inch stem, a few of them. That to me is winterization worthy. And
I'm just trying to figure out if you give GA, you can get stem elongation. If you have an active bud, you can get elongated stem, but if you need new buds, then you should be probably spraying with a cytokinin.
yeah,
And do they have buds? That haven't grown, do they have buds, but they just haven't broken and grown, or there's just no buds?
I am assuming until March, that they all have potential buds, if not real buds.
So
And-
just trying to point that out.
Yeah.
And if you spray GA before bud break, they'll break.
Which actually would not be a bad thing.
And I know Brandon usually uses GA47, which is the advantage of using GA47 over GA3 is that it's metabolized, and so it doesn't have a it doesn't have a continual effect over a long period of time, which
Okay,
can be a detriment if you don't want the stems to keep growing and growing and growing and growing. So,
okay,
So you're saying, if you don't want the stems to keep growing and growing, you use GA47.
because it goes to the normal GA oxidation pathway,
Yeah.
and so you can get a quick effect, but it doesn't give you, there's a lot of, in the nursery industry, there's a lot of reasons why they want things to grow, but they don't want them to keep growing, become long and spindly.
Yeah.
And so using GA47, it's much more popular in apple shape than it is in grape bunch, but it's pretty cheap. It's not as cheap as GA3, which is dirt cheap. But
okay.
anyway, but Brandon will tell you.
Sure.
He's done that. He has papers on that.
Yep, and he said in an earlier email thread that kind of relates to him receiving this information and my asking that, do you want to be involved in it? He said, yes, definitely, but, so.
Yeah, I just wanted to make sure you weren't trying to induce new shoots off the main shoot, in which case, I said kind of.
I just ask a lot of questions to a lot of experts. And the 3 or 4 kernels literally go into test. And that's about as far as I can take it. So.
I used to go to a lab and it had a sign on the wall, which fits me quite well. It said expert knows more and more about less and less until you know everything about nothing. That's me.
It's a very fine point. And then related to that, Lou Gerton and I have talked quite a bit about other biostimulants or inhibitors to try to balance what they do over the course of 6 months to get poinsettias ready for a 3-week window. Ethylene for BRAC development and a whole bunch of things that can potentially be applied here. So the toolbox is open and we're just trying to get all of the cells and numbers and all that kind of stuff set up. Couple other feature data points that we could put in test. I personally am interested in the endo and ectomycorrhizal effects. Protective calcium, maximizing uptake, those kinds of things that will enhance the volume of my project, not necessarily the health, but in some ways it's the same thing.
So using mycorrhizae in the potting material of the stock plant.
Yes,
Okay.
and in the stick potting material too.
Mhm.
So as they are getting the first adventitious root out, that they have a symbiotic
Mhm.
helper.
Does Gerton already use that kind of system with other species?
So
So
let me put it this way, not with perennials. I think they
Okay.
use it in regular greenhouse,
Yeah.
but not in a big way.
Uh-huh.
And so when I've talked with them about this, it's kind of like, well, yeah, we can test it,
Behind time.
but it's not a commercial standard that they are working off of.
So they don't mind using biological, the whole thing about sterile soil. As long
Not,
as it's known or assumed to be beneficial, it's okay.
yeah.
Right, and I think we should maybe address the question in the chat
Okay,
and then formally wrap up, because
Sure.
we're over. So there was one question about cutting hazelnut stems as dormant cuttings in March or April before the buds open, noting that willow stems cut in March are very successful. Has that been tried?
we're not gonna do any hardwood cutting, which I think that addresses directly. We're only doing softwood, green tissue. Even some of your earlier questions with email about, semi-hardwood.
Right.
I think we may have gotten some of those, but I was in that second cutting that I did. Knowing what that looks like, but my intention was only to cut with softwood.
I did some hardwood cutting trials some years back, and time of collection was one of my variables. I don't remember if I tried that.
Okay,
If I did, it didn't work, but if I didn't try it, maybe it should be tried.
Okay.
I'd have to look back at that paper.
General, the rooting rates on the hardwood cuttings is not very high, or hasn't.
And operationally, if you cut one woody stem and try to get it to root, you're missing 10 other softwood cuttings that could come out of that healthy stem over the next 3 months.
Sure.
So it's a reduction in potential volume, and I gotta steer away from that.
Yeah. Yeah, great. So that was a good answer. And we had another, usually Lee posted saying that she's interested in feeding both substandard hazelnuts and hazelnut waste to pigs. And folks on the chat have already mentioned that Jason Fishbach would be a great source for those things. So we'll make that
What
connection.
was the name of that?
Usually Lee is a researcher at Morris. She will be presenting in this lab meeting discussion in a couple of months. So about other hog feeding trials. So that could be a cool co-product market access option. All right, thanks, everyone. Great presentations. Obviously, we can't stop ourselves from talking about hazelnuts, which is a good thing. And we'll see you all next week.
Thanks, Steve.
Sure.